Okay, so again, just to kind of look at what we're discussing here, we're going to look at the tissue type, right? And characteristic of muscle tissue, and then the function of muscle, right? And yesterday, what we discussed is the types of muscle versus cardiac versus smooth, right? So we saw that table there. Characteristics of muscles, which we covered as well. So we talked about the 4 characteristics that make muscle tissue very the main functions of muscle, and then we talked about some of the other characteristics, for example, like connective tissue that's found with muscle. Again, these are both connective tissue, right? But you've got muscular tissue and connective tissue here, and this is how we organize it. And we use these words, epi versus peri versus endomyosin, to describe the divisions or the sectioned off areas by the connective tissue that a muscle like this would have, right? So if we took looks like a femur here, and we took the biceps femoris, which is your big old fat muscle on the front of your leg, make your quads, then we would take that big muscle and then cut it in half, and you would actually see these individual kind of looks like groups here in muscle. And in fact, that's just one of those right there, right? We took one section and pulled it out. And we took that one section and looked at it, and we call this a fascicle. And we took that fascicle then and we pulled out one muscle cell. All right, so this is what you see right here is one muscle cell. Just to show you how it's all grouped and organized together. And now what we're discussing of that, right? And I multiply that by however many there are here, however many there are here. You can already see that if you do it all together, this whole muscle will make a movement. Does that make sense? What you see is the muscle getting short, like long here, short there. But in fact, it's occurring cellularly and molecularly, individually. Sarcomere, 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 all of them doing it. When you do them all and all, this is that muscle doing this. Does that make sense? We have to fill in the gaps though. There's more information to talk about. But that's kind of, you got to step back and look at the big picture, because that's the big picture right there, right? We're going further in, but then we're like, what does this mean? You got to step back and talk to yourself. To where these two start to touch the M line, and that's kind of where we can fully contract. We can't go any further. Okay. Z discs, for example, will never touch each other, by the way. There's a limit to muscle contraction. Okay, so we are stepping back even further. We looked at how contraction kind of works, but we're going to step back even further. So we're kind of going back to the beginning, about talking about who's doing the decision making to tell muscle, okay, time to contract, right? And again, the ultimate operation station headquarters is going to be the brain, of course, right? Brain is going to make a decision, time to contract muscle. Signal is going to be going down the spinal cord. It's going to be sent to neurons, and we call these motor neurons because those are going to send signals to do some kind of action, right? Which means muscle contraction here. We know that neurons are neuronal tissue. That's the nerve cell, right? Neuron. Neurons and muscle cells have similarities in the fact that they both are excitable cells. They can undergo an electrical signal, in fact. And we talk about that being a membrane potential voltage, right? It's a membrane potential, or it can generate electricity, in fact. We know a nerve does that, but muscles also do that. The nerve transmission, or the nerve signaling in a neuron and a muscle cell is called a action potential, or AP. Okay. Now, how do we get the neurons signal to the muscle, for the muscle to start another signal? We do that through chemicals, obviously. And the chemical we use to transmit that information, we call that a neurotransmitter. It comes from the neuron. We call that acetylcholine, or ACH for short, acetylcholine. Okay? Action potential is the signal. Neuron and muscle have to interact with each other. They have to, neuron has to tell muscle cell, I want you to contract. So the signal comes down to the end of the neuron. And the way they talk to each other from that point is the nerve sends a chemical out, and the chemical is called acetylcholine. Acetylcholine binds to the muscle cell, and then means to the muscle cell, okay, we're going to contract. Okay? That's what we're seeing here. Now, when we talk about membrane potentials, or generating an action potential that we're talking about, this electrical signal that occurs in nerves and muscle cells, we do this through a series of changing ion concentration. When we change ion concentration, that means we let ions come into the cell or ions go out of a cell, and that's going to change the membrane potential and generate an electrical signal. Because if I separate that's my crude little phospholipids, right? If this is my plasma membrane, okay, and I physically separate ions that have this charge and ions that have this charge, I've generated potential energy, okay? The positive represents ions that are positively charged. So we got to go back to chemistry, right? Unfortunately. That means an element that has lost an electron. Remember that whole thing? You lose an electron, you become positively charged. You gain an electron, you become negative charged. So if I'm this ion, I'll erase that so it won't be too close. If I'm this ion, and I'm located in this area, I exist in the world as a positively charged ion, which means Na plus means you've lost an electron. Does that make sense to you? If I'm this, then I exist in the world gaining an electron and I feel fine. this all goes back to that rule of 8, right? Octet rule, remember that whole thing? Stability. And we know that sodium by itself, without it losing an electron, has 1 in its outer shell and needs like 7 to become stable. And so rather than doing that, it gives away the electron, becomes stable. This one has 7, it needs 1 more. And so naturally they get together, they're always kind of hanging out together. It gains an electron, becomes stable, but we use a negative sign to say that it's gained an electron, okay? Now if I separate it like I'm doing here, okay, and then at one point I say, you know what, I'm going to open up a channel, remember integral proteins, right, and cell membranes, and open up a channel and allow these to flood into the inside of a cell. When they do that, they generate energy. Energy in the form of an action potential or an electrical signal. Okay? And this is how the nerve sends the signal down itself. Okay? Now, 2 ways you can actually open up channels in a membrane. Depends on what cell it is. You can have a chemical open it up, or you can have electricity open it up. Okay? Okay? Chemical can do it, or electricity can do it. We call them ion channels because that's the channel that just allows ions. We're not talking about anything else, just ions going through. Okay? Those are really important to understand because we're going to get to that point where we see how they're put into the whole step process. We're just seeing 2 different ones. They actually look different themselves, but you're not going to see a figure in it in the lecture exam that says, tell me what this one is or is that one. You need to understand chemically gated ion channels opened by chemicals, and in particular, when acetylcholine gets released, the muscle cell plasma membrane, the sarcolemma, has receptors for acetylcholine, and when acetylcholine hits, that's what tells the gate to open, to let stuff go through, and how the electrical signal will generate in the muscle cell. Towards the end of the neuron, or the nerve cell, we've got these channels that when electricity hits them, they will open up. All skeletal muscle will get stimulated by somatic motor neurons. Those are neurons or nerve cells that are specifically for the body and specifically for motor responses, which means telling muscle to contract. That's the effector, right? Axons, which are parts of the nerve cell, okay? It's an extension of the nerve cell for these motor neurons, go from the central nervous system to skeletal muscle, which means that one nerve cell starts at the spinal cord, and its axon or its extension can travel from the spinal cord all the way to the skeletal muscle. So some nerve cells can be as long as, I don't know, a meter, a little over 3 feet long, traveling down your body, right? Now, once the axon hits the end of the nerve cell, it will branch. The ends of it will hit the nerve fiber or interact with the nerve fiber. And we call that forming a neuromuscular junction. Or motor end plates. okay? NMJ is for short, right? Neuromuscular junction or motor end plate. Each muscle fiber, each muscle cell in every single skeletal muscle has one neuromuscular junction and one motor neuron feeding it. Which goes to show you that there is a lot of motor neurons, right? A lot of motor nerve cells. So this picture does, figure 9-7 does a great job of putting together where you would see a one motor neuron coming from the brain and really through the spinal cord, and that's where it begins, right? That neuron begins at the spinal cord level. But we know that there's many neurons connecting spinal cord to brain and all in between. So there's neurons and neurons and neurons, and they're all interconnecting, making decisions, and they finally send that signal to one motor neuron there. And that motor neuron's cell body, where you find the nucleus, is kind of close to the spinal cord. Its extension that we call an axon travels from there all the way through the body to, let's say, the biceps brachii muscle. Because that'll be our example of a skeletal muscle we're going to contract. When it gets towards the end of its neuron, it's going to split. Can you see how it's split? 3 there at least? But there can be more than 3. It's many branches. Each one of those branches will hit one muscle cell. This is one muscle cell. That's another muscle cell, muscle cell, muscle cell, muscle cell, muscle cell. Inside of it are those rods, the myofibrils. We know that myofibrils are made of sarcomeres. So you can you don't have to just imagine how many sarcomeres all lined up next to each other for just that one bicep. The interaction between the neuron and the muscle, where the neuron ends and the muscle interacts with, is called a neuromuscular junction. That's one. This is one neuromuscular junction. There is another neuromuscular junction there. There's another neuromuscular junction there. But all individually spreading out to different muscle cells. Because muscle fiber means muscle cell. I know I keep mentioning that over and over, but you have to put that in your brain because you will forget it. You'll think of fiber or something else, and it's actually a cell, right? The end of the axon is called the axon terminal. And between the axon terminal and the muscle fiber is a small space called the synaptic cleft. Okay? Stored within the axon terminal are these synaptic vesicles, and in these vesicles, these are inclusion bodies, right? And these vesicles are a collection of acetylcholine, that neurotransmitter, that chemical, waiting for direction or stimulation so they will go to the edge of the axon terminal and release the neurotransmitter. But it's not going to do that unless it's stimulated to do it. Does that make sense? It's kind of waiting there in these vesicles. Only when the nerve sends its signal down is where they're going to migrate towards the end and release out the neurotransmitter. On the other side of the sarcolemma, on the muscle fiber, we have these folds called junctional folds. And in those junctional folds, we have a lot of receptors for the acetylcholine. Those are chemically gated ion channels, right? right? We just saw a picture of a chemically gated ion channel. But that's on the muscle side. And all this stuff here that we're talking about all together is part of the neuromuscular junction. Okay. So let's take a look at a neuromuscular junction. This is our typical neuromuscular junction, right? Notice that we just see a portion. Everything in yellow is the neuron or the nerve cell, but it's only a portion of it. We don't see the long tube that's the axon. We don't see the airy beginning of the axon. We don't see any of the rest of the neuron, just the end of it. Because that's what we're caring about right now. That's what we're looking at in particular. You see these little circles, these holding chambers, which is plasma membrane basically kind of just in one little bubble. And inside of it, these green yellow dots. Green yellow dots are the neurotransmitter acetylcholine, right? And obviously we've already stimulated the acetylcholine, the vesicles to migrate, because we're now seeing what happens when they migrate or move towards the end, the axon terminal, and we see what happens when they go to the end. They release out the acetylcholine. We see the acetylcholine going and migrating across the synaptic cleft, the space in between, and interacting with the acetylcholine receptors on the chemical gated ion channels. And we see what happens when the acetylcholine binds to the receptor. We see what? The gate open. You can see them like this, right? There's at least 4 there. 5, sorry. Over here. And what is that doing? Opening up, letting ions go through from here that are here inside. Not the green things, ions that you don't see here. That's going to generate this electrical signal down here. There'll be another electrical signal forming here. And that electrical signal will go down the sarcolemma, down to the T-tubules. Remember I told you what the T-tubules are, right? Stimulating those channels on the SR to release calcium, to start contraction. Okay? Here. Okay? Kind of pink, whatever, orangey, whatever that color is. First, the events at the neuromuscular junction. Okay? Action potential arrives at the axon terminal. The action potential is already generated by the neuron. And in the nervous system chapter, we'll discuss where that, how that occurs with them, but we just need to know that the signal's already come. The decision's been made by the brain through the spinal cord to tell our muscle, okay, it's time to contract. Send that signal down the neuron, the nerve cell, to then do that. So it arrives at the very end of the neuron that we call the axon terminal, little bulb kind of end. What we haven't seen, this is another detail, is that action potential stimulates these channels there called voltage gated. That's the key word, right? Voltage gated. Calcium channels to open. And an ion that we call calcium floods in to the axon terminal. Calcium ion causes the release of the neurotransmitter or neurotransmitters into the synaptic cleft. What it is doing, folks, in number 3 is that calcium has stimulated the synaptic vesicles to migrate and release acetylcholine out. That's what calcium does. Okay? Acetylcholine diffuses across the synaptic cleft, gets received by the acetylcholine receptors on the acetylcholine chemically gated ion channels on the sarcolemma on the muscle side. The binding of the acetylcholine to the receptors causes the ion channel that opens up for sodium to enter into the muscle cell and then causes an end plate potential or action potential in the muscle cell. I'm sorry. To be generated. Okay? Now, the number 6 one talks about an enzyme that gets released when acetylcholine binds to the receptor. We call that acetylcholinesterase. ACE means enzyme. And what this enzyme does is it breaks down acetylcholine. So what I drew there is just a channel that has acetylcholine on it that the channel's open. Everybody see the channel opened, right? So what we're seeing here, if you're looking at what's coming in inside the muscle, would be a bunch of sodium. And it's generating the action potential in the muscle going that way, right? Everybody get what I'm saying here? Okay. What acetylcholinesterase will do, this enzyme, will go in remove that acetylcholine from the receptor and break it apart. Degrade it, right? Call it degrading, but break that chemical apart. When that happens, then the channel closes. No sodium goes in. The process will stop and it's ready to go again. Does that make sense to you? It's only when we remove these and break them apart is only when this channel will close back. Acetylcholinesterase has to be released to break that. Break it up. Here's what we're seeing, that 4 steps I just mentioned are kind of what we're seeing here. It's just extra stuff here. If you notice the pink arrows up top, the pink arrows up top represent the action potential arriving, stimulating the channels that calcium will, the channels that will open that allow calcium to go through, stimulates these synaptic vesicles to migrate and release acetylcholine. Acetylcholine releases and diffuses or spreads across the synaptic cleft to the receptors. Acetylcholine binds to the receptors. The channels open up. You can see the channel open up there. Okay. Action potential will be generated here inside the muscle because this is muscle over here. That is a neuron there. That little white line represents the sarcolemma or the plasma membrane of the muscle. And we see that signal getting generated down here. Then we see what acetylcholinesterase does. You see that little enzyme there, and it breaks acetylcholine in half. You see it breaking in half. Removes it from the receptors, and there you see the channel closing. Okay? So what about this? Myasthenia gravis, right? Here's the characteristics. So you look at, the pathogenesis here, right? Drooping upper eyelids, difficulty swallowing, talking, muscle weakness, generalized muscle weakness. Well, why do you see that? Because there's a shortage of acetylcholine receptors because the body's attacking the acetylcholine receptors. This is an autoimmune disorder or disease. For some reason, the immune system thinks that the acetylcholine receptors in somebody that has myasthenia gravis are foreign. It says you're not supposed to belong here. So as I mentioned, sometimes the immune system goes haywire, right? And it thinks it's it's an enemy, it's foreign, it's not supposed to be there, and starts attacking it. When it attacks the receptors, then there's no place for acetylcholine to bind. Thereby, the channels don't open up, the signal doesn't get to the SR, the calcium doesn't get released, actin doesn't bind to, or myosin doesn't bind to actin, contraction doesn't happen, the muscle does not function, and that's where you see weakness. Myasthenia gravis, this is what occurs. Okay, let's talk about this, right? Here's the generation of the action potential of the electronic signal in the sarcolemma in the muscle. So we're talking about muscle cell. Occurs in the muscle cell. Sarcolemma means muscle plasma membrane, right? The sarcolemma in resting state, which means we're not generating a signal or nothing, we call this a polarized state, which means that the plasma membrane is holding out all the other ions and creating this difference. Positives on the outside, negative on the inside. This is polarization, right? Or polarized. The inside is always negative compared to what's on the outside. And this is normal. We haven't done nothing yet. Okay? We want to look at when the neuron decides to be stimulated to send acetylcholine over to open up the channels. What's going to happen then, right? So this is the before, and this is going to go the what's going on, right? The end plate potential is generated. We get depolarization and repolarization. So let's talk about that, okay? The release of acetylcholine binds to the receptor. The chemically gated ion channels open, letting sodium go into the muscle fiber. The inside of the sarcolemma becomes less negative or more positive, however you want to think about it. This difference here, the electrical signal that's getting generated because we change the ion concentration inside the cell, is called the end plate potential. And now we're at the beginning stages of the generation of the action potential or the electrical signal inside a muscle. So that's what you see there, right? If you look closely, you can see that the channels have opened up. Sodium's flooding in. The end plate potential is getting generated. And we're going to do something with that pink line. The next part is called depolarization. The generation of the action potential. As more and more and more sodium floods in initially, we'll get to a point where it will cause the neighboring channels to open up and really flood in sodium. That point is called a threshold, where when we hit the threshold, those voltage gated sodium channels will then open up and it will really flood through. Okay? This triggers then the action potential because now a large opening, all the channels open up around it, causing a large flooding of sodium, causing then an action potential to get generated. This spreads across the sarcolemma from one voltage gated channel to the next and to the next, and will spread out. The action potential will spread out. This is generating the action potential. That action potential will finally get to the SR to open up the calcium channels, or the channels to open up to let calcium go through. This is what we're seeing here. So we see this box right here, and this box represents those sodium channels, right, opened up. So we have to hit a certain number of ions coming here, sodium ions coming in here. We'll call that a threshold. And once that hits there, it'll spread across, hit the neighboring sodium channels, like that purple one, to cause all of them to open up, and then really generate what we call an action potential, or AP. That AP will be unstoppable. It'll cause the other channels next to it to open up, and cause the channels next to it to open up, and it goes down the line, spreads across the whole entire membrane, or the sarcolemma. And that action potential will continue and continue. If you stick a probe in there, let's say you stick a really precise voltmeter, right, measuring electricity, and you stick it into the muscle cell like this, you would read electricity, right? Really, really, really small amount, but the electricity, this is what we're talking about, electricity will be formed. That's an action potential. Number 3, we call this repolarization, but it's resetting, okay? Resetting, getting everything back to normal, back to the original state, back to the resting conditions. So eventually, after the action potential is spread, those sodium channels close, and another channel opens up, calcium channel. But that calcium channel will allow potassium to exit the sarcolemma, bringing down the concentration back to normal, where we saw positives on the outside and negatives on the inside. Okay? So these potassium channels that open allow ions to escape, bringing it back to a normal range. And then there are some other channels called the sodium-potassium pumps that finely tune or adjust the concentration consistently. Those will pump everything back and forth because they have the capability of pumping stuff in and out, and they will do this. So here's our third figure right here, right? We're just adding to the whole entire thing. But you see now the sodium channels are closed right here. And potassium channels are open, letting potassium go out, getting everything back to resting state. We call that repolarization, back to normal. Now this thing can go all the way and start all over again, depending if the nerve wants to send another signal, and then contract it again. Talk about measuring voltage or electricity. We can measure it, right? So this has been done before in the research, right? We see that when the channel's open, there's a big change in electricity, right? You see the graph go up when the sodium channels are open, depolarization. When the sodium channels begin to close, guess what happens? Everything kind of goes back down. Electricity will go back to normal. We saw that repolarization. And here the graph continues to just show you that we're in a resting state. So excitation contraction coupling is what happens when the action potential that travels along the sarcolemma is coupled with contraction. So in this event, we see the action potential stimulating through the T-tubules, the SR, and their channels to release calcium. Because calcium, when it releases, will cause actin and myosin to bind together or form a cross bridge. This kind of middle step between what would just happen, the action potential forming in a muscle, to where calcium gets released and a contraction happens. This is a middle stage. Okay? So here's what occurs, right? We're just kind of stepping back. We're kind of going a little bit further back just to see the big kind of what's occurring. And you see the motor end plate or the axon terminal. You see the acetylcholine getting released. You see the little receptors there binding and opening up, channels getting released. You see this big pink line on the right and the left means the action potential. Action potential travels down the T-tubule. You see one T-tubule, you see those little pink arrows that go down the T-tubule. It stimulates all the channels there next to the SR to release and release and release. Open, open, open. And those little tiny dots that are kind of, I don't know, red, brown, whatever they are calcium. And those will diffuse to the thick and thin filaments. That's where they're going, by the way. This is internal. This is inside, right? Well, now we see a close view of the T-tubule and the SR and the other SR. We call those terminal cisterns. And all three of them is called a triad. This is the triad, right? The three. You see calcium, the channels opening up. Calcium released. Calcium will diffuse to the actin, myosin area. And what do they do, right? Calcium binds to troponin, causing troponin to change shape, moves tropomyosin, the little line, out of the way, exposing the binding site. Then to allow myosin heads to bind to actin. So calcium releasing causes tropomyosin to move out of the way to expose the binding site. That's its function. Does that make sense? And then everything automatically happens. Head will use energy to bind because it doesn't need anything else but to move that little site out of the way, and it's like almost like a magnet. We're not using magnetism, but kind of like what you would see if a magnet does that. And bind, and then pull, right? They start doing that little ratcheting thing. The thin filaments get closer and closer together, contractions occurring. So nothing can occur, again, if the neuron doesn't fire, if acetylcholine doesn't get released, if an action potential, if the sodium channels don't open up, if action potential doesn't get made, if that doesn't come to the T-tubules, if calcium doesn't get released, none of that can occur. If none of that occurs, then we can't have myosin heads bind to actin. It can't happen. Okay? So again, low concentrations of calcium, real low concentrations. Tropomyosin is going to block the active site. Heads can attach. Muscle fibers relaxed. It's only when we increase the amount of calcium because we released it is when we see this cross form, right? Calcium binds to troponin. Troponin changes shape, moves tropomyosin out of the way. Head binds to actin. Cross bridge forms. And then heads start moving because of ATP, energy. Okay? when we stop stimulating from the nervous system, when acetylcholinesterase breaks down acetylcholine, when that stops, closes the gates, everything gets back to normal. And what happens with calcium is it also gets pumped back into the SR. So calcium gets reused again. So SR can pump calcium back, thereby releasing it from troponin. Tropomyosin will slide back and cover the binding site. Heads will detach, and the sliding filament thing will slide back. Little thick and thin filaments will slide back. All right, so what we're looking at here, cycling, where we form the cross bridge, the heads pivot and pull using ATP. They cock back, come down, back if they want to, detach and start again. Let's talk about rigor mortis, right? So rigor mortis happens after somebody dies, right? This doesn't necessarily need to be just humans, because animals can do the same thing. 3 to 4 hours after death, we start to see a stiffening of the muscles. Okay? Body begins to be hard, stiffen up. That occurs for about 12 hours post-mortem. And what's happening? Calcium cannot be pumped back into the SR, and so it continues to leak out. As long as calcium is there in the sarcomere, heads will bind to actin and pull, and then not detach. This is just to show you that if we pump back calcium, then the heads will detach and troponin and tropomyosin will move back into place. But since there's no energy being produced to pump calcium back, calcium stays on that site. Heads stay there. Vis-a-vis your body after 4 hours after death stays stiff. Does that make sense to you? Now, after that 12-hour span, what will occur is that the body then becomes flaccid. It loosens up. Okay? Muscles will loosen, will get soft. What's occurring is that the proteins that make up the thick filament, the thin filament, the dystrophin, all those proteins we just talked about that make what it is, break down. They break apart, break up in pieces. So the sarcomere begins to fall apart. When it falls apart, muscle becomes relaxed, and that's where the flaccidness. So we see this weird thing where 3 to 4 hours stiff for 12 hours and then it not stiff. Right? That's where